Hello everyone. Today's chain seminar is 34th chain seminar. We are welcoming the Daisuke Shimaoka from Monash University. Daisuke did a PhD in 2012. Yeah, he completed PhD in 2012 in University of Tokyo. We actually in the same university, same department, in different lab. And then he started a postdoc in 2013 in University College London. And he continued working there until 2019. And he's now a research fellow in the biomedicine discovery group in institute, Monash University. So today he's going to talk about neural dynamics in terms of the encoding property and also the stochasticity of the brain dynamics. So please, stage your listen.
Thank you, Keisuke-san, for kind introduction. Today I'd like to talk about the dual nature of the brain processing. And so my research has been trying to understand how the brain processes visual information. For this to happen, the brain somehow represents a visual information with neural spike trains, and the spike trains are transmitted to other brain areas. And studying this process is extremely hard because it involves many organs, such as retina and dozens of brain areas involved in vision. We cannot simply isolate one of these components, but have to treat them as a whole. In such a circumstance, one way to approach is treating this as a sort of a black box, where our only measurable inputs are the visual stimuli and the observable outputs are basically spike train. So my talk today is all about opening this black box and sort of reverse engineering the brain, particularly visual cortex, to understand the input-output relationship. So in the field called systems neuroscience, the neuronal response is basically described in this kind of framework called linear nonlinear model. There's no need to understand the detail here, but the point here is that there are two essential aspects of the visual information processing in the brain. The first part describes how a neuron responds to a visual stimuli. So neurons in the visual cortex are not simply responding to visual stimuli, but they respond to specific attributes of the visual information. And after this initial process, the input is passed to a stochastic nonlinearity and then to a stochastic spiking component shown here, which then results in spike trains that are observable to us. This second component is supposed to be complete random to explain the variability in observed firing. That is why this component is represented as a dice. So today I'd like to speak about these two aspects of the brain processing in vivo. Firstly, I will talk about the problem of encoding, particularly focusing on so-called higher visual cortical areas using marmoset monkeys. And secondly, I will briefly talk about the apparent stochasticity using mice, not just visual cortex, but across almost the entire cortical hemisphere. So let's start with part one. So now let me speak a bit more on the visual system. So photons hitting the retina is converted to spike trains there, then transmitted to a part of the thalamus called LGN, then to a primary visual cortex or B1. From here, what is believed is that the visual signal is transmitted successfully to other visual areas. And if we look a bit more closely, the visual information is initially transmitted from B1 in the cortex, then to B2, and then to B3, and then diverged into two streams called dorsal and ventral streams. So the visual system in the cortex is organized as a hierarchical system from lower areas such as B1 to higher areas, higher visual areas. And it is believed that the higher visual areas process more elaborate visual information. Then how does each visual area process visual signal? As I mentioned, the input-output relationship in each area is described in this framework of linear nonlinear model. The first step involves convolving the incoming visual signal with something called receptive field. But it's simply the receptive field refers to the area in the visual field that affects the response of a particular neuron. And each receptive field has a particular preference to a specific visual attribute. In this particular case, shown here, this neuron prefers an elongated vertical bar. And by the way, this idea of receptive field is not limited to the visual neuroscience, but also to other modalities such as audition and smart sensation. Now, let me show a typical procedure to estimate the receptive field in an actual experiment. So here, an animal, it's actually a cat, was presented rows of white and black bars on the screen under general anesthesia. And at the same time, an extracellular unit recording was conducted from the primary visual cortex. And every time the spike occurs, which are sometimes shown here, the visual stimulus preceding the spike event was identified, and its position in the visual field is registered in this window. This is a case for the black stimuli, and this is a case for white stimuli. And this is a combination of the white and black. After many repeats of the same stimulus presentation, you get an estimate of receptive field, from which we understand this neuron actually prefers vertical stripes with white and black colors side by side. So this procedure of estimating receptive field works extremely well in an early visual areas such as retina, LGN, and B1. However, over the years, it's also becoming clear that this approach does not really work in higher visual areas. There are a couple of potential reasons for why the conventional approach was not successful in higher visual areas. First, higher visual areas encode increasingly more elaborate visual features, but covering all the combination of stimulus increases rapidly and becomes intractable. And second, in this approach, it is essential for experimenters to make sure that neural activity is not induced by other factors, such as animals' movements, because that's going to be a confound. To minimize the confounds, animals are often immobilized, often by general anesthesia or by physical restriction or by behavioral training. It remains possible if these highly regulated unnatural conditions can impact neural firing. So to overcome these limitations, here I apply another approach, which is called encoding modeling approach, proposed by Dr. Nishimoto in Osaka University nearly 10 years ago. This approach uses more naturalistic conditions, such as presenting natural movies and behavioral conditions where animals are allowed to move by their will. And to these observed brain signals, such as spike trains or MRI data, we fit a model of the brain so that it matches the observed signal. And this is done with machine learning techniques. And after doing that, we inspect the refined model of the brain to examine what the model encodes about the explanatory variables. Here I'd like to talk about the application of this approach in two of my ongoing projects with marmoset visual cortex. So the first part is about third order visual areas, or sometimes called B3, located here in the diagram. So this is a bit of detail about the possible organization of the B3. So despite extensive study of primate visual cortical areas, the functional organization of the third order visual area remains actually still elusive or contentious. There are two competing hypotheses, and these competing hypotheses propose a single, just one third order visual area or two third order like visual areas in the marmoset cortex. The former hypothesis predicts that the dorsal portion of the brain is covering lower portion of the visual field, whereas in the other hypothesis, the dorsal portion of the brain actually covers a portion of area preferring the upper visual field. And one of the difficulties in studying the third order visual area is that it is presumably a quite small area, particularly in marmoset, and its width is just about 2 millimeters. So hitting this area with a tiny electrode is not that easy. And despite this small size, we still need to reveal the functional organization within the area to judge the areal boundaries. In other words, the recording technique needs to cover a large field of view with high spatial precision. So to achieve this, I combined two recording methodologies. First, I developed a so-called wide field intrinsic optical imaging for marmoset with a rather big cranial window. So this is an opening on the skull, about the size of 10 by 13 millimeters, that covers multiple visual areas like B1, B2, B3-like structures, and even higher visual areas. By the way, this wide field intrinsic imaging involves illuminating the cortical surface with visible light and measure its reflectance. So the resulting optical signal reflects changes in blood flow caused by neural activity. So it's kind of similar to functional MRI. But this imaging has much higher spatial resolution, so it has been used to study sub-millimeter structures within an area, such as cortical columns. And secondly, following the wide field imaging, we also conducted a single unit extracellular recording using a high density probe called the NeuroPixels probe shown here. This probe is about 10 millimeters long, along which 384 channels are recordable at once. This electrode was carefully inserted which is shown here. So inserted along the cortical surface to cover as many areas as possible. And during these recordings, we presented a variety of stimuli, such as conventional bar stimulus and patches-like stimulus, in addition to the naturalistic movies. And to analyzing the imaging signal with the encoding modeling approach, we adopted a motion energy model. This model is composed of more than 6,000 linear filters or kernels, and each kernel covers different portion of the visual field with different orientation and spatial frequency. And we fit all these kernels with the observed imaging signal in each camera pixel. Now let me show how this fitting would look like in just one pixel shown in this portion of the brain. This fitted model captures the signal fluctuation, which is shown here as a purple trace. So it captures not everything in the observed signal, but some portion of it. And as a result, the correlation coefficient between the fitted signal and observed signal is about 0.3 correlation. And as I said, there is more than 6,000 kernels we estimate, and their coefficients are distributed not evenly, but rather in a skewed manner. And if we look at just the top 6 kernels, then they look like this. Sorry, they are not very visible in the screen, but all these 6 kernels are preferring this portion of the visual field and somewhat different spatial frequency and orientation. And with the fitted kernel coefficients, we can run in silico simulation to obtain the location of the receptive field, which is shown here in this particular example. Now, we repeated the same fitting procedure in every camera pixel. And if we zoom out from a single pixel to a wider range, we can see that the individual pixels prefer slightly different portion in the visual field. And these positions, preferred positions in the visual field, is now color-coded in these 2 panels. So the left one is altitude or elevation, preferred elevation. The right one is the preferred azimuth or x-axis. So these positions in the visual field, as you can see, the preferred position gradually shifts across brain areas, meaning there is a retinotopic organization. Notably, we observed a limited portion of the cortex, shown as red here. This portion is actually encoding the upper visual field. And also bear in mind that in the very anterior portion of the image region here, this model fitting did not go well, as quantified by explained variance. And this indicates that the estimated preferred position in this brain region is not very trustable. We were able to confirm our imaging result also with the electrophysiology in the same animal. So both EFIS and imaging resulted in pretty much the same preferred altitude. So these imaging and EFIS results strongly support the hypothesis that third-tier visual area is composed of actually 2 different areas. And the area borders are defined objectively by computing chirality of the preferred position called field sign, which is shown here. And this result is nicely recapitulating the previous result obtained by my colleague Liz Zabitz. In her case, she obtained this map using a 10 by 10 array electrodes implanted into 4 different marmoset monkeys. But this time, we were able to show the same result from the single animal. And we were able to confirm this result also in the other 2 animals in the image in the same approach. So to summarize this part, the data-driven modeling applied to the marmoset mesoscale imaging was able to identify retinotopic organization up to 3rd-order visual areas. And it supported the multiple V3 hypothesis. However, our investigations so far also indicate a limit in studying even higher areas adjacent to V3 shown here. This actually leads to another investigation I'm talking about. So the dark area which we just saw is called posterior parietal cortex, or PPC, which is actually known to be a multimodal area. So the PPC, among the PPC is actually can be divided into multiple areas. And one of the area called lateral intraparietal area, or LIP, is known to respond to both visual stimulus and also to eye movements. So in the last part I just talked, all the recording was done under anesthesia. So this means that animal did not move their eyes. And maybe this was the reason for our failure in the last section. So in this part 1, 2, we conducted extracellular unit recording in the awake condition from chronically implanted chamber and a linear electrode from 2 marmoset monkeys. I will today talk about a result from just one animal, because we basically replicate the same result from the other. So we conducted recording during a Q-saccade task that involves both visual stimulation and animal's eye movement. The gist of the task is 3 components. First, a dot appears on the center of the screen, to which the animal directs its eyes and keeps fixation. And second, a face stimulus, a face of the marmoset, which we call a target stimulus, is presented somewhere in a periphery in the screen. And third, the animal directs its eyes to the target to obtain a liquid reward. During this recording, we continuously monitored visual stimulus positions, eye speed, eye position, and pupil diameter and blinks with an infrared camera. And during one recording session, the animal typically performs hundreds of trials. And most of the cases, animal can successfully direct its eyes to the target, but in a minority of cases, it fails to direct its eyes, usually just keeps fixation. And moreover, there are abundant periods of intertrial interval in which animal is free to move its eye by their will. And the animal also blinks during intertrial intervals. So during all these stimulus and eye movement related events, a single unit emits a series of action potentials shown in the bottom. With these data at hand, we aimed at studying the 2 essential aspects of the multimodal encoding. First, what aspects of eye movement related signal is encoded in LIP neurons? Second, whether encoding of LIP neurons are consistent within the tight control of eye movement, and also during the free viewing condition. To answer these questions, we again took the encoding modeling approach, this time with the generalized linear model. We designed a model to house 5 linear temporal filters or kernels corresponding to the 5 modalities. And to be a bit more precise, each kernel is combined with the time series variables of each modality to reproduce the observed spike time series. Now, an obvious question is whether all these kernels are necessary, particularly eye speed and position kernels. To examine that, we constructed another model without eye position kernel and compared it with a full model with 5 modalities. So it's pretty clear that this reduced model explains consistently less amount of the observed data compared to the full model. And likewise, the reduced model without eye speed explains less amount of data compared to the full model. So these results indicate that both eye speed and eye position information are necessary to explain the observed data. The last slide was at the population level across 600 recorded units. Now let me turn to individual units. Here I show 3 example units during the successful trials with presentation of the target stimulus, shown as red vertical line, followed by eye movement or saccade to the target. All these 3 neurons appear to respond to the stimulus that are well captured by our full model, shown as blue traces. Sorry, not really visible on the screen. But just computing the stimulus-triggered average does not tell us whether these units are encoding visual stimuli or eye movement or something different. So here, the power of our approach is that it lets us to decompose the observed signal into components encoding different modalities. So to decompose the observed signal, we use only a subset of fitted kernels while ignoring other kernels. So after this decomposition procedure, the red trace here represents the model response using only the stimulus kernel. The dark and light green traces are model response with the eye speed and position kernels, respectively. In the first example unit shown here, this apparent response is actually mostly driven by visual stimulus, but not by the eye speed or position. In the second unit, the response is dominated by eye speed kernel. In the third example, its response is actually composed of stimulus, eye speed, and position kernels, almost equally. We can confirm what these neurons encode by analyzing the data when the animal did not saccade to the target stimuli. In this condition, the stimulus evokes firing to the first unit and third unit, but not to the second unit. We can further confirm the encoding by looking at the data when the animal makes spontaneous eye movements in the absence of visual stimuli. The spontaneous eye movement evokes response in the second and third unit, but not in the first unit. And by the way, these 3 conditions are sampled from different conditions. The first condition was sampled from within the task and during the successful trials. The second condition was from the failed trial when the animal kept just fixation and did not saccade to the target. And third condition was sampled from intertrial intervals. So this means that our model captures the firing behavior in multiple situations within the successful and failed task trials and also outside of the task. So these decomposition analysis reveals individual units are actually driven by different modalities. And these differences can be quantified with explained variance computed only with stimulus kernel and explained variance computed with the x-axis, with eye speed shown in the x-axis. In the first unit, the stimulus kernel explains most of the observed data, so can be called as stimulus-driven unit. The second unit, on the other hand, its explained variance is dominated by eye speed, so this can be called as eye speed-driven unit. In the last unit, explained variance is about the same between the two, so it can be called as an integrator of the visual stimulus and eye movement. At the population level across 600 units, the contribution of each modality are distributed rather continuously, showing heterogeneity of encoding across units in LIP. Finally, these are the kernels for pupil dilation and blinking from individual unit and their average. The pupil dilation kernel shows somehow interesting oscillatory behavior, and this waveform is remarkably consistent across recorded units. The eye blink kernel is somewhat more variable, but still shows kind of stereotypical waveform around time of blinking. To summarize this part 1, 2, the encoding modeling approach with generalized linear model revealed much broader encoding of individual neurons in LIP. First, LIP neurons encode information on visual stimuli, eye speed, and pupil dilation and blinks. And among these, contribution of visual stimulus and eye speed and position differs greatly across recorded units. And secondly, LIP neurons fire consistently during tight control of eye movement and also during free viewing. To summarize the part 1, we have seen the sort of power of the encoding modeling approach. Firstly, applied to the wide field imaging in anesthetized higher visual areas, results from this approach supported the multiple V3 hypothesis. And applied to the unit electrophysiology, this approach revealed encoding of visual stimulus and eye movement information in a single unit scale. Now I switch my focus to my second of my talk for the last 5-10 minutes. So far, we have assumed that neurons in the visual cortex process the same visual information, always in the same manner. In other words, we treat the neurons as a deterministic system. However, this is just a gross simplification. In reality, when presented with the exact same stimuli, neurons in vivo generate quite variable responses. And to account for this apparent variability, the standard linear nonlinear model posits stochastic spiking component. This stochastic output is widely believed to infer from the brain's so-called ongoing activity that happens in the absence of sensory input. So the question I want to address here is whether the ongoing activity is completely random, as the standard model postulates. Now, let me start with a wholesale record of a single unit, single cell. That this was recorded from the mouse parietal cortex. The membrane potential of a single neuron sometimes shows sporadic transitions between depolarized and hyperpolarized phases without presence of any external stimulation. And these phases are sometimes called up and down phases. Now let's look at these up and down transitions at larger spatial scale, such as across different parallel fields. And each barrel encodes different whiskers in rodents. Just as in the left figure, we record membrane potential of a single neuron from this portion of the barrel with a glass pipette. And at the same time, the population level membrane potential is measured with imaging fluorescence of voltage-sensitive dye with a camera. So the neuron located here shows ongoing transitions as shown as black traces, and the simultaneously recorded optical signal adjacent to this neuron tracks these up and down transitions as well that are shown as red. Now, if you look at the optical signal representing membrane potential across this entire field, the depolarization signal is actually a wave propagating from one barrier to the other. In this case, it propagates from left to right and then to the top. This was done like 20 years ago, but now the technology advanced, so we can actually look at even larger scale. When we further zoom out and optically monitor the almost entire part of the cortex from both left and right hemispheres, we immediately notice that this depolarization event is actually at the scale of the entire cortex and travels across many areas, including visual cortex. So these studies illustrate that ongoing activity that looked like a random noise at a single neuron level is actually not a complete random, but has some sort of structure in space and time, and which can be only captured by recording at wider scale. So in one of my previous studies, we noticed this depolarization wave is not always the same or not always different. Rather, it looks like that wave elements have some sort of repertoires or propagation pattern. So this recording was done when the mouse was deeply anesthetized and monitored until it recovers from anesthesia. And like in the last slide, we optically measured mesoscale voltage signal. And the average signal across the entire cortex shows ongoing up and down oscillations, as you can see. Furthermore, in this particular event, and if we look at each individual part of the cortex, the wave propagates from left to right in this particular event. And in this case, it's actually kind of opposite, from right to left. And in this case, it's from bottom to top. Through the hierarchical clustering analysis, we found 3 typical wave patterns across all the animals we investigated. This could be more easily visible just by showing the average events across many events. So these results, together with previous studies I talked about, it is now becoming clear that ongoing activity is not just random, but has an order in space and time domain. In the subsequent study, we did the same wide field voltage imaging, but this time when the animal is fully awake and spontaneously ran on a spherical treadmill. In this condition, the ongoing activity is completely in a different regime. There is no wave propagation across cortical areas, but instead we found that spontaneous running induces depolarization in an area-dependent manner. So in areas like motor cortex and auditory cortex and primary visual cortex, we saw depolarization. On the other hand, areas in the S1 barrel cortex and areas called LM, which is a homologue of B2 in Lorentz, we saw hyperpolarization. So to summarize this part 2, we found that the cortical neurons, even in the sensory areas, have a very rich intrinsic dynamics at cortex wide scale. More specifically, the mesoscale ongoing activity has a structure or order in space on the cortex and also in time. And this spatial temporal order can change by the arousal state of the animal. So this is a conclusion slide of my talk today. So the overall picture I want to present is the sort of the duality of the brain processing. In the first part, we went through that how neurons in the visual cortex encode inputs like visual stimuli and also eye movements. And the encoding modeling approach was a powerful technique to reveal it. On the other hand, in the second part, we went through that neurons in every part of the cortex has its own spatial-temporal dynamics, which is behind the apparent stochasticity of the cellular firing. So an emerging picture from these studies is that to understand input-output relationship, these two aspects are essential. In future, I'd like to deepen this view, for instance, by including this stochastic component part into the generalized linear model to refine the model of a single neuron or a region of or a bunch of neurons. Thanks for your attention. Happy to take questions.
Thank you, Shimaoka-san. If you have any questions from online, you can raise your hand, or we can also take questions from the lecture room. Can I just have a quick question? Because I'm a bit, I'm not expert in this area, so especially for the first part.
Yeah.
When you reveal the kind of encoding pattern in briefly and stuff, I'm just not sure like how much new your result and how much that can contribute like applications. Is it applicable for like BMI and stuff like that, or like what is I just didn't get like how much it wasn't clear and how much your research the new evidence and how that maybe I just didn't get that kind of how to contextualize your findings.
Yeah, so that's a good question. I think the primary novelty of this research in the first part is that our results supported the kind of contentious hypothesis about the organization of the B3, which is shown in the first part. And as for the second part, my understanding is that previous studies has revealed encoding of each different modalities, but not necessarily the combination. And so this comes to the next possible answer or applications. So this kind of approach can be extended for combinations of many modalities. And I think there is a kind of way for possible application in this direction. And even in more naturalistic cases, like even without any physical restraint, like free moving in the cage or something, for instance.
Okay, thank you. Any other questions? Jason.
Thank you for the talk. My question about second part or part one, especially on the popular diameter and its destination. So I'd like to, I'm wondering if you could give us more interpretation of that result, because popular diameter is regulated. I really heard that popular diameter is regulated by the total Lafayette, and it's related to reward expectation. So if the FYP for therapy is protecting the eye movement and visual information, but also it's like coming information from the reward expectation, the motivation experience, it would be very interesting for me.
That's an interesting comment. I mean, I have no concrete answer, but I think we could incorporate, like reward expectation in the model, and we might be able to study whether such, reward expectancy might be encoded in LIP by extending, this approach.
Thank you.
For part two, stochasticity.
Yep.
So if your traveling wave happens random, then stochasticity is not inconsistent with spatial temporal.
Yeah.
Because your traveling wave is really uniform and velocity.
Mhm.
So, yeah.
Yeah, I mean, that's right. I don't think people has really studied on this aspect. I mean, people just treats this stochastic part as complete random without actually studying its structure.
The randomly happening traveling pulse, traveling wave there, then it's suggested and special information.
Right.
Is this like that or some other complex patterns there?
I'm pretty sure there are by now. The study by after me explored many more patterns actually. Yeah.
So the pattern generated or suggested also there.
Yeah. So in our hand, with imaging data, we managed to find 3 typical propagation patterns that we found in every mice we studied. There were actually quite a lot of kind of inter-animal difference, which we actually did not really talk about in the paper, but I'm pretty sure there are more patterns.
So I'm not sure if I thought the stochastic response is the typical in the brain.
Yeah.
But in your work, the important thing is that the stochastic of the propagating pattern is important one.
So we were actually trying to find the order between the patterns. Like we were thinking about the possibility that some pattern like this may come more often than other patterns. But in our hand, we didn't really find it. So the short answer is we couldn't find it in the order at the higher level. But maybe the way we studied might be optimal to find that order.
And also, why do you use the anesthetic state, anesthesia state? Now, I thought the recover from the anesthesia state. Anesthesia state. There are lots of paths from the unconscious to conscious. I thought. So it might be because of that, it can be regarded as a stochastic response. But if you use the static stimulus to the mouse or something, then I thought we could see the stochastic response of patterns of propagation. But why do you use that?
We were interested in how the depths of anesthesia change the brain dynamics. And what we found, which I did not talk today, was that this pattern itself is actually conserved when irrespective of deeply anesthetized or nearly awake. So that spatial temporal pattern itself was conserved. However, we also found that the speed of propagation is rather accelerated towards awakening. So yeah, in that sense, there is a kind of interesting dynamics in that scale as well.
Thank you. And also the first part, the models.
Yeah.
And actually, I didn't understand what is predicted by the model.
So the model was, maybe I can speak a bit about how we fit the data. So basically, we just try to fit the this time series data.
Which part?
So in this case, it's this is just a single unit spike traces.
So which part of the brain?
Oh, this is from an area called lateral intraparietal area. So located here.
And a single neuron.
Yeah, that was a single neuron.
So what happens if you change the neurons? So maybe next to this neuron, and then if you do the same thing, then the same kernel will be selected one.
Yeah, so that's exactly what I did here. So these 3 units are recorded from different portions of the brain. I mean, all from LIP. And there are actually quite a lot of diversity across recorded units, which is shown here. So one dot is basically one unit.
So unit means the single neuron and not the place.
I mean, precisely speaking, we can't make sure that it's really a single neuron, but that's why we call it a unit. But right. Yes, go ahead.
I'm curious about this arousal.
Yeah.
Arousal versus NSTs, the different scarcity. Can you somehow bridge this result to like IIT integrated information stuff, or do you think it's related or not? I assume you're also interested in this kind of.
Oh, sure. I mean, why not?
But I mean, I remember like Massimini study, like when they do the TMS, they just propagate the brain
Right.
way
Yes.
more when you are awake compared to the.
Yeah.
Is it kind of similar things happening?
So if we try to connect to that kind of studies, as towards awakening, actually, apart from these global patterns I have talked about, there are more kind of localized patterns. So in that sense, as you approach towards awakening, the spatial temporal pattern can be more complex, and maybe that's related to what Massimini had done.
I know it's kind of impossible to calculate phi in after brain data.
Well, I mean, in theory, you can approximate.
Yeah.
But what I heard from Nao's chair was that computing phi in this high resolution is not practical for now. In future, it might be, but not now.
Anything else? Any question from online participants? No question from the audience. Yeah, okay, then thank you again. And we haven't yet decided the next chain meeting, but yeah, we will announce in the website. So thank you very much again to Daisuke. So
Thanks for inviting me. It's really my pleasure to be here. And thanks for your attention to the Zoom audience.
okay, I'll stop Zoom, and if there's some people in chain is around in university, we can actually have some drink together and use your place. Thank you.
Oh.